Most eukaryotic cells follow a process of growth and division called the cell cycle. These events include a growth stage, mitosis or nuclear division, and cytokinesis or division of the cytoplasm. Throughout interphase, the cell is engaged in growth and metabolic activities. Interphase can be further broken down into three discrete phases called G1, S, and G2. During the G1 or first growth phase, normal cell functions occur as well as cell growth. In the S phase, DNA replicates, producing 2 copies of each chromosome. During the G2 phase, the cell continues to prepare for mitosis and cell division. Mitosis, or the M phase, has 4 stages: prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes become visible and condense, becoming shorter and thicker. Each identical copy of a single chromosome is called a sister chromatid. The nuclear envelope breaks down and spindle fibers form as microtubules grow out of the centrioles that move to opposite poles of the cell. During metaphase, the double-stranded chromosomes line up along the equator of the cell. The spindle is now fully formed and the microtubules attach to each sister chromatid. Anaphase begins when the sister chromatids of each chromosome begin to separate. The centromere that holds sister chromatids together divides, and the chromosomes move away from each other along its spindle fiber. In telophase, the 2 groups of chromosomes reach the opposite ends of the cell. As a new nuclear envelope starts to form around each group, the chromosomes uncoil and the spindle disappears. The division of the cytoplasm and organelles is called cytokinesis, or the C phase. The result of mitosis and cytokinesis is the formation of 2 genetically identical cells. To summarize the entire cycle, G1, S, and G2 phases occur during interphase while the cell is growing and preparing to divide. Mitosis, or the division of the nucleus, consists of 4 phases: prophase, metaphase, anaphase, and telophase, and in these phases duplicate copies of the chromosomes are split equally. Finally, the cell's cytoplasm divides during cytokinesis and results in the formation of 2 genetically identical daughter cells.
This objective began with a short video. Please, I would highly recommend watching that video several times in order to memorize and become familiar with the detail. Objective 1 asks that we list the stages of the cell's life cycle and be able to describe what occurs at each of these stages. Most of the cell's life is spent in the non-dividing state called interphase. In body cells or somatic cells, there are 3 stages in cell division. There's DNA replication, where the genetic material is duplicated exactly. You have mitosis, where that genetic material is divided equally. And then you have cytokinesis, where the cytoplasm and everything else in the cell, the organelles, is divided into 2 daughter cells. Interphase is characterized by 3 phases. G1 phase, you have cell growth, you have duplication of organelles and protein synthesis. S phase is DNA replication and histone synthesis. And then G2 phase is when you have the finishing touches. Proteins are continuing to be synthesized as well as centriole replication. Here we see an image from the textbook that represents interphase, and you can read the details. But G1 is normal cell functions plus cell growth, duplication of organelles, protein synthesis. S phase is when the DNA is replicated and you get synthesis of the histones. In terms of DNA replication, there are a number of enzymes that are used in this process that I'd like you to know. The first one is helicase. Helicase, and remember ASE at the end of any word tends to represent an enzyme. It's the role of helicase to unwind the DNA strands. If you remember, DNA is a double helix, and if you were to straighten that out, you would have a ladder-shaped molecule. And the role of helicase is to unwind or unzip those two complementary strands. DNA polymerase then runs along these individual strands. So here's DNA polymerase, and it's going to run along the strand, and it's basically going to build a complement. Let me see if I can easily change colors. So for instance, if the nitrogenous base here is A, it's going to build a complement to that, and the complementary base for adenine is thymine. If this is C, it's going to put a G next, so on and so forth. And it's actually going to do that for both strands. So what you end up with are one and two strands, so two identical strands of DNA. And that's the role of DNA polymerase. Lastly, ligase will piece together any sections of DNA that may have a gap. So for instance, it's not uncommon for gaps to be created, and it's the role of ligase to actually come in and piece this back together to fill in any gaps that exist. So these are the enzymes that play a role in the S phase of interphase, where DNA is being replicated. Here we see that process in action. Here we have helicase unwinding the two complementary strands, and then you have DNA polymerase right here in step 2. These large blue spheres represent DNA polymerase, and it's going to run along those complementary strands and build a complement. So in the end, you end up with two identical copies of DNA. Step 3, again, DNA polymerase is running along the strand building. And then lastly, step 4, DNA ligase is going to fill in any gaps that may exist. Eventually, you end up with two identical strands, as we already noted. G2 is characterized by just the finishing touches of cellular duplication. So you have protein synthesis, you have centriole replication occurring, continued cell growth, and then we move into mitosis. And mitosis is characterized by 4 phases: prophase, metaphase, anaphase, and telophase. As a student, I always remembered PMAT for those in order. During mitosis, you divide the duplicated DNA into 2 sets of chromosomes. The DNA coils tightly into chromatids, and chromatids connect at a centromere. Often it's represented by sort of an X-shaped molecule with the centromere at the middle. So you have individual chromatids that are then joined at the centromere. In addition to that, there's a protein complex around the centromere called the kinetochore. I often think of these as molecular handles, and this will become apparent in a minute. In mitosis, we start with prophase. The nucleoli disappear. The centriole pairs move to opposite poles of the cell. The microtubules extend between the centriole pairs. The nuclear envelope disappears, and the spindle fibers are going to connect to those kinetochores. Remember those molecular handles, kinetochores. The spindle fibers physically connect to those little handles. During metaphase, the chromosomes align in the central plane called the metaphase plate. Here we see that happening. Here we have prophase. So you get a disappearance of the nuclear envelope. You get the centrioles moving to opposite poles of the cells. You get the formation of the spindle fibers as they reach out and connect to the kinetochores of the located on the centromeres. And then we move into metaphase, where you get an alignment of the chromosomes along the metaphasial plate. Anaphase, the microtubules pull the chromosomes apart, and the daughter chromosomes group near the centrioles. And then lastly, telophase. The nuclear membranes reform, the chromosomes uncoil into chromatin, the nucleoli reappear, and the cell has two complete nuclei. Here we have anaphase. You can see the individual chromosomes being pulled apart towards the opposite ends of the spindle apparatus. And then telophase, once you have nuclear division, telophase begins cytoplasmic division. Often the formation of this cleavage furrow is a clear indication that a cell is in telophase. And then cytokinesis would be the physical separation of that cytoplasm, of those organelles. And that's going to give rise to two identical daughter cells. So cytokinesis, division of the cytoplasm, the cleavage furrow around the metaphasal plate, the membrane is going to close, producing two identical daughter cells.